Mentioned, I'm Aida Ebrahimi, Associate Professor in Electrical Engineering and Biomedical Engineering at Penn State University. So today I'm going to share with you our work on real-time label-free electronic monitoring of bacterial conjugation using graphene microchips. So a lot of you perhaps have heard about antimicrobial resistance, or AMR, which is one of the major health issues of our century. So basically, it's a natural behavior of bacterial cells that they acquire resistance against antibiotics. And throughout the years, the number of deaths caused by AMR has been increasing. So every year, more than 1 million people die directly because of AMR. And by 2050, it is projected that more than 10 million people will die directly because of AMR, which is actually going to be more than the total death combined of HIV, cancer, and cardiovascular diseases. So it's a major problem. One of the main mechanisms that bacterial cells, they have developed throughout, like basically since Earth started, throughout billions of years, is through this mechanism that is called heterogeneous gene transfer, which happens basically when two bacterial cells, a donor and a recipient. The donor has the resistance gene in it. They conjugate, they form these pili, which are basically protein bridges. They can range between 2 micrometer to 20 micrometer. And within a couple of minutes, basically, they transfer the gene to the recipient cell. And that's how they basically give the disease to the next bacteria. And then they multiply, right? So there have been imaging techniques that have actually shown this in action in real bacterial cells using confocal laser microscopy. You can look at this image, these videos. So basically what will happen, the donor cell, they will extend the pili, and then they will actually so let's look at this video, the image part of it. So they start, they extend the pili, and then there is going to be attachment, and then they pull each, they get actually connected. So the conjugation will happen, and then they release. Okay, so there is this action in population of bacterial cells. So the current methods that are used to study this, which is, by the way, very important to understand how cells transfer the gene resistance genes and understand the dynamics of this phenomena, which can help us to develop better therapeutics, especially when we have biofilms and we have co-culture of different bacterial cells in biofilms, that there is a competition between this cell and that cell, how they transfer these genes. Understanding of these mechanisms are very important in our effort to actually address the AMR issue. So there are methods that are conventional biologists use, like PCR, we are familiar with it, which is an endpoint method that you basically get the cells, get the genes out, look at what you have in different cells. But it's an endpoint method, and it's very tedious and labor intensive. There are also imaging techniques like the previous slide from the PNAS work that they showed. Imaging, so they use microscopy, great tools. However, they require labeling, which again, they actually can impact the biology of cells themselves, and they complicate the system. And one of the problems with microscopy systems is they have limited field of view. So this AMR gene transfer that I showed, it's a population phenomenon. It's not, it is single to single cells, but because the frequency is a stochastic mechanism, in order to understand what is the, let's say, statistic or probability of this happen, let's say if you have this population, bacteria X versus population bacteria Y, you need to actually be able to screen a large population, okay? So we need to have a wide field of view, and microscopy cannot do that to really get the resolution that we want using it videos because of the field of view limitation. So there is, to the best of our knowledge, there was no, at the time that we had this proposal, and then we got the funding from NIH, there was no method that could actually do continuous label-free direct monitoring of the dynamics of this conjugation. And so our idea, proposed solution was basically to do electronic readout. So use graphene chips, make basically a chip that can do large area, like as we all are familiar in integrated sensor community. So we can build chips quite easily, and then monitor spatially the signals. We can monitor electrically. And then our idea was, if you have bacteria cells functionalize on graphene channels, basically, because of this pooling factor, which causes a shear force, mechanical force, this pooling that I showed in the video, as you saw, they literally, like from 20 micrometer distance, they get to 0 distance. So just imagine like the mechanical force that it's going to actually cause. So we were thinking, can we actually use graphene because it has a high ion module and it's a flexible material? Can we use it to tether the cells and then look at the resistance change and the fluctuations, basically, and from that get the frequency of this and monitor this phenomenon in real time? Okay. So why graphene? It has a very high ion modulus and very high carrier mobility, which gives us high sensitivity, basically, and its resistivity can be modified, or its piezoresistive behavior, basically. It can be modified by some defect engineering as well. And the gauge factor that is 2 to 3 times more than normal metals, and it is generally very biocompatible, and it is also optically transparent. So if we want it, we can actually do at the same time optical image. People have done optogenetics using graphene nanoprobes, so it's optically transparent, unlike let's say gold metal and stuff, etc. It's not fragile, so it was a good substrate to actually use for this application. And we can functionalize it quite easily using some chemistry, like, for example, if we use P-based chemistry using pi-pi stacking without any covalent bonding, we can actually functionalize graphene. So we basically, this is the fabrication method. We use epitaxial monolayer graphene. So they are a little like, they are monolayer, but they have a buffer layer that kind of sustains them. So we have a 2D-CC, a 2D material consortium at Penn State that they basically were able to modify the graphene, the layer number, the doping concentration. So we explored quite different varieties. Then we etching graphene to isolate like different graphene channels. I won't have time to go through like the design of the channels, but we looked at different designs so we can get the maximum sensitivity that we can. Because like I said, it's going to be resistance based. So we want to, with those tiny force that is going to be applied, we want to have maximum change of resistance that we can. So we looked at different designs as well. And then like putting electrodes, all of them with photolithography and passivation using aluminum oxide, then like, exposing and making devices. I mean, you cannot see them very well. You have to turn the lights off, but there are graphene channels. The channel width is 5 micrometer. The strip is, so there are strips like this, basically, and the gap between them is 2 micrometer, I believe. Yeah. So yeah, so it looks something like this, but there are arrays of them. So basically, we use P-base to as a cross-linker. Use the antibody for E. coli. E. coli was our model, by the way. So which is the same model that was used in the Clark paper, in the PNAS paper. So it was a good model, already validated. So we use the same. We follow the same protocols. We did the engineering of the genes. So we had receptor bacteria as well as receptors that they didn't express these pili proteins. So they were knocked down. So and then we had donors. So they were some engineering, which my colleague in food science, they did that part of it. And like using BSA blocking the non-specific bonding, and then applying the donor plus recipient or recipient-only control bacterial cultures. And then at 37 degrees C, which is the usual temperature we look at bacteria culture. So we continuously monitor the resistance. And by the way, the donor strains, we validated them using PCR as well as some optical imaging. They expressed 1,000 times higher frequency of conjugation, which was consistent with the Clark paper, the PNAS paper. So this is some SEM images. So the bacteria, they, as we wanted them, they actually are functionalized on the graphene stripes only. And so we confirmed the functionalization step at different, like from the pristine graphene to P-based treatment to P-based antibody. So we consistently see a P-doping from Raman spectra, which was expected based on P-based charge. And we looked at AFM to again confirm how these different layers are actually functionalizing graphene. So as we, like from graphene all the way to antibody, the height difference was like, captured using both using AFM and from molecular perspective, Raman confirmed it. So, and then we have imaging. So then we have the chips. We wire bonded them on our chip carrier. Use the DAC system to monitor the resistance values, and in our temperature and humidity control system, we monitored as bacteria. Basically, we basically incubated the bacteria, receptor plus donor, and receptor only, and looked at the resistance using the DAC. So here is what we saw. So pre-incubation, these different steps. And so I will go back and show another slide that shows different steps better. So all the way before, there is only there is no bacteria. So as you see, the noise of the resistance is pretty much like the same. But once we added the cells, we saw significant change of the noise of the resistance when we had donor and recipient cells compared to only recipient cells. So recipient cells, there is no possibility of generating those pili. They cannot express them, okay? And donor receptors, the donors, they shoot them out, and then there is a chance, it's a stochastic phenomenon. There is a chance that they're going to conjugate hit receptor, recipient, and then they're going to pull them. So just imagine you have these cells here, and they are like doing this. So pulling and like releasing until they lock. And that's what we actually see. There is a significant noise variation of the resistance that we are measuring. So this is like ABC, these are pre-incubation at the steps that we actually did the functionalization steps. Remember, we have graphene, we add P-base, then we add the antibody, then we add BSA, right? This step is the same no matter what bacteria you have. So we expected at that stage, and we were continuously functionalizing and monitoring, okay? At those stages, we don't expect any difference between the two populations because there is no bacteria introduced yet. Then into the our chamber, we introduced the bacteria and then kept monitoring. So then this is just like, another way of looking at the overall trend from the previous signal. So basically using this technique, we showed proof of concept of how we can use these graphene-based chips to do label-free direct monitoring of bacterial conjugation in real time. Basically the mechanism is that this conjugative interaction, they create this shear force on the underlying graphene, and those, because graphene is a very good piezoresistive material, they translate in the resistance variation that we could observe. We have spent a lot of time to make that work. It went through 2 PhD students to actually make it work. I'm finally it did. Now we actually are working on doing simultaneous electrical measurement and optical measurement, and using other better piezoelectric 2D materials, not just graphene. The good thing with graphene is it is carbon, it's very chemically inert, and it is very stable. Other 2D materials, we have to be very careful. Okay, I don't know if you're familiar with the field. They are very good if you have a miracle sample, but they have like variability, so you have to be careful. So we are working with the 2DCC Center, which they are actually focusing on large area materials. So they have had very good samples, and we are actually extending our work to molysulfide and doped molysulfide and other 2D materials, which actually have bandgap. They actually have shown better piezoelectric behavior and even piezoresistive behavior. Piezoresistive behavior, of course, piezoelectric behavior. Those are 2 different things. And also, so we did time result analysis, but we want to do a spatial result analysis with a better resolution. And with that, I have to thank the students, especially Vinay and Derrick. So the two of them, they are the first co-authors co-first authors of this work. They really did a lot of work, and the funding agencies. Thank you very much.